Welcome to this, well, let me start again since we just started the live stream. So welcome everybody again for this last day of lectures. So it has been a long marathon so far, and we see the end of this nice school, which I really enjoyed. 1 hour and a half break, and then we're going to have one panel, two lectures, and one last panel. So without further waiting, I'd like to leave the floor to Vishu to share his screen and start the presentation. So thank you very much, Vishu, for being here.
Hi everyone. Are you able to see my screen?
Yes.
Okay, excellent.
Is not okay. Yes.
It's now full screen, right?
Perfect.
Okay, great. Thanks everyone again for joining me today. And I know it's the end of the third week of the long workshop. I hope I had great fun, and I know I really appreciate every one of you who has sort of been hanging on and attending the last day of the lectures as well. So since the last day, I will try to sort of go slow and try to be interactive. So if I do not if I'm going if I'm not clear about anything, somehow I don't get any sort of message when I sort of when there is a question over the chat box. So maybe somebody can prompt me, or just unmute yourself and speak. So that's also fine if you have a question that you would like to ask. So today, in the third talk of the series of talks I am giving, I'll talk about how intrinsic noise that is sort of within the system, sort of and the bistability interact with one another in very interesting ways in animal collectives. So, of course, in the last talk, I spoke to you about this self-organization patterns in plants. So these are collection of plants, and today I'm going to focus on animals. In plants, what the fundamental principle I sort of mentioned was that there are these feedbacks between plants that cause different types of patterns. For example, this simple example of local positive feedback that decays to zero after a finite distance, that will give you irregular patterns where the cluster sizes are not same. On the other hand, if we had a local positive feedback followed by a short range negative feedback, then this leads to periodic regular patterns, which are also very popularly known as Turing patterns. Okay. So likewise, there are these animal patterns, and I also spoke about bistability and noise. I know this is just to remind you, because I will make some connections to this in the end. So we thought of bistability as a potential well and a ball rolling in this potential well with two minima. And system can go back and forth between two minima. This just like the ball, can bounce back and forth between two minima because of stochasticity. So just to remind you the basic concepts. So, but today I'm going to talk about noise, bistability, and patterns in animal collectives. And to do so, I will start with this video. I hope this plays well, and I hope my network is not too bad. Fish, krills, and many other species, you will also find these kind of patterns in cells, bacteria, where as an aggregate, they are performing extraordinarily synchronized motion. And what you're seeing in this specific part of the video is how a simple computational model can reproduce some of these observed patterns. So again, this is actually a simulation. What you're seeing right now is a computer simulation, and how it can fairly accurately describe the observed patterns. So, and here is an example of a collective motion from the laboratory where we work. where we have just placed a whole bunch of fish in a fish tank. So these are about 750 fish in this specific case. Again, without any stimuli, even in the experimental conditions, we actually can observe these kind of collective motion and synchronized movement patterns in animal groups. So, so the sort of broad outline for my talk today, with that motivation of couple of videos, is to understand how do animals exhibit this collective motion, and what are the basic mathematical and computational models that signal in. I'm going to sort of explain that in steps, mathematical theory behind this, what are the methods and experiments that we have done to sort of show that. And if I have time, I will also talk about some of our ongoing work on dynamics of collective motion under predation. Okay, so when we think of these animals in groups that are moving, people ask sort of two classic questions. One is that of ecological or proximate question. How do organisms exhibit such remarkable collective motion patterns? So to sort of break this question further, what is it that each animal is interacting with other animal, other organism, that produces these patterns? Okay, that's the basic mechanistic question that one is interested in. And secondly, one can also ask, why do organisms exhibit collective motion? What are the costs of being in groups? What are the costs of moving in groups? At the same time, there has to be some benefits that animals gain by being in groups and moving in groups. How does the trade-off between these costs and benefits sort of weigh? So those are two broad class of questions people try to understand in the field of animal behavior. So today I'm going to focus only on the first question. How do organisms exhibit these patterns? More specifically, how is each fish or each bird sort of talking to or interacting with its neighbor and producing these patterns? So here is a sort of a classic model of collective motion. So it's some of the early models of collective motions were developed by computer scientists and physicists in 80s. Those are known as the Boyd models, for example, developed by a computer scientist, Derek Reynolds. They're also widely used in animation industries. And later on in 90s and early 2000s, there were more sophisticated, more detailed models from a physics point of view as well as biological point of view. So here, the model I'm going to present today is from Cousin et al., 2002. So here, basically the idea is we can represent these groups of fish or insects or birds as actually point particles. Although I'm showing you cartoon pictures of fish, you basically assume in the model that these are all point particles, but with some rules. And what are those rules? There is a tendency for animals to exhibit local repulsion, meaning if two fish are too close to each other, they repel from each other in their movement. And secondly, once they're not too close to each other, they exhibit these two social interactions. One is that they have local attraction with each other, and that causes cohesion. And they also have local alignment with each other, that causes directional alignment of the entire group. And one can write down equations of motion for each fish in these groups. Okay, and then if you sort of iterate these rules over using computer simulation approaches, what you find is that animals may follow these very simple rules like local attraction to stay cohesive and local alignment to stay in the direction of motion of neighbors. They can actually show striking patterns. So here is an example. So depending on this, so I spoke to you about two important forces or interactions, right? Attraction and orientation. For example, if the attraction is very, very strong and the orientation is not very high, you often will find a cohesive sort of group of animals, but they don't have directionality in their movement. On the other hand, if the orientation is the dominating force, if the animals are strongly aligning with each other, then what you find is they not only form groups, they also move in a polarized fashion in some arbitrarily chosen direction. And finally, here is something very interesting that's very hard to guess. If there is some balance of this attraction and orientation, you find patterns like this, very counterintuitive. Why do I say it's counterintuitive? Because there was nothing in these rules of attraction and alignment that tells them to go in circles, right? Yet, if you put them together, if every individual fish, every individual insect follows these rules with some strengths of attraction orientation, you can also find them just going in circles. Okay, so basically these kind of models can not only explain observed patterns of collective motion, they also make predictions about new forms of motion that were not very obvious. And in fact, there are many organisms that show this kind of milling behavior. Okay, so those are the simple, I have just introduced you to one simple class of models. These are also called self-propelled particle models. And in this context, okay, there are all these models. How do we know? How do we know they're right? How do we know what are real organisms doing? Are real organisms following these simple equations of motion? Are they really exhibiting these local alignment, local attraction rules? Uh, but to address this problem is really challenging, right? Because imagine I observe flocks of starlings or schools of fish, hundreds of them, tens of them. By observing them or by recording them, how can I really infer what's happening at the local rules? It's not very obvious, right? And to counter this problem, people have devised experiments with very small numbers. So I'm going to give you a very brief review of this. So this was one of the very first papers trying to understand how animals move in groups. These are what they did. This is a group in Rome, which I'm sure many of you might be familiar with. And what they did was they recorded and they sort of took very high resolution images of starling flocks. And what they did was, from these images that were taken from multiple cameras, they were able to reconstruct the positions and the orientations of each starling in the flock. And from analysis of the positions and a quantity called anisotropic factor. So I'm not going to explain what exactly this is because that's a bit technical. And what they basically conclude are two things. First is that the starlings interact with one another not based on distance, but rather based on who are their some hierarchical neighbors. For example, what they found was they interact with 6 to 7 neighbors independent of how far they are. So the distance doesn't matter, but what really matters is that they are interacting with, on an average, 6 to 7 neighbors. And once you and with that kind of a inference, they were able to sort of reproduce many of the observed patterns in the model, in the data. And there were a whole series of other work where the so I'd see at one extreme here you have large number of starlings that are moving, and from them you infer what they are doing. The one other extreme is that do experiments in the laboratory, put only a few number of fish, 2 fish, 3 fish, maybe 5 fish or 10 fish, and from the movement of those fish, people have tried to infer what are the rules that animals are following. So there are many such approaches. I'm not going to go to a whole review of this, but the sort of summary of many of these studies is that the simple physics inspired self-propelled particle model that I mentioned are not really what the animals seem to be doing. Animals seem to be doing something sometimes often much simpler, sometimes much more complex. So animals are probably doing something fundamentally different. So nevertheless, there are some challenges in understanding what are the rules at the level of individuals. For example, if you were to do experiments at the level of only 2, 3, or 4 fish, you have inferred how are they interacting when they are in 2, 3, or 4 fish groups, right? However, how do we know they will be true even when I put in 25 fish? Maybe the rules of interaction among fish will change as the group size increases. because the density increases, maybe they have to do something different to stay cohesive and stay polarized in their direction of motion. So it's not very evident whether these rules will scale to larger groups as well. And secondly, many of these studies sort of ignore the role of stochasticity in how interactions happen. So, it's also not very clear whether the inferred rules are unique. For example, the more detailed observation you make, sort of, you will find a lot of, interesting but detailed behaviors. But the question is, are they important? What do they really tell us? Obviously, there will be differences among fish even within a species, and there will be differences among individuals from one species to other species, and of course, naturally across taxa. So so it's not very, so one must really aim to understand some basic underlying principles of this collective motion. Okay, and in the context of understanding how animals interact, it also really depends on the scale that we are studying and the context we are studying. For example, if my question is what makes group cohesive, I will get some set of rules that keeps animal groups cohesive. On the other hand, if my question is what makes group motion highly polarized and directional, and that will determine, that will give me a sort of maybe a different set of rules, right? And moreover, the ecological context matters. For example, are these animals facing predation, foraging, and that again might determine the types of interaction animals are following. So in this sort of broader context, the focus of my presentation today is a recent work that we have done, where we try to understand the dynamics of group polarization. The first thing we see when we observe a group of animals moving is how nicely polarized they are, and how sort of spontaneously they change these direction of motion, and there is some element of stochasticity, and there's some element of determinism in them, right? How do we understand this dynamics of polarization? So given that the focus is on polarization, I'm going to focus mostly on deciphering the rule of directional alignment in collective motion. And in doing so, I'm going to hopefully convince you that the role of noise, one must not really ignore noise, and there is very interesting signatures that the noise can provide. So how do we do this? I'm going to first describe the experiments we did. Okay, so what we did was we took a fish tank. With shallow width, and what that really helped us was, because it's a shallow depth water, fish were effectively swimming in 2 dimensions, and therefore we could record all the group members from a camera mounted on the top of the fish of this tank. So here is the picture on the right shows a snapshot where we have observed the fish school is somewhere near the center. They're all moving towards the right part, and then we use techniques of image processing to identify the coordinates of each fish, as well as the direction of motion of each fish. And this inset here shows the colors show the trajectories of each fish over a short duration of time. And the fish we are using is called karimeen. It's a fish that is native to southern part of India. Okay, so here is a video, I hope it plays, of how these fish schools look like. I'm showing you a group size of, I think, 60. Okay, you can see how they're mostly cohesive. Occasionally there are some individuals who move away, but on a longer time scale, those are really rare events. And we do these experiments for about 1 hour long, and we have done these experiments for 15 individuals, 30 and 60 individuals. And then as I explained to you, we did what's called image processing, which is basically by the fact that between 2 frames fish have moved some distance, we can construct the direction of motion. And once we have done, we know so on this slide, focus on the top right corner here where I have showing VI hat. So VI hat is the direction of motion of the fish I at time Tn. And if I know the direction of motion of each fish, I take the vectorial average and we get something called a group polarization, M of Tn. So what is this M? If I look at the modulus of this vector, if this vector is 0, the fish are moving in random direction. On the other hand, if the modulus of this vector polarization is close to 1, that means that fish are nearly all aligned with each other and they're moving in the same direction. And you do this analysis for every frame of your video. Actually, we don't do it for every frame. We skip a few frames in between because sometimes fish just don't move because between consecutive frames. So from that, we calculate this time series of polarization as a function of time. We are showing you here only for 6 minutes, but this is roughly, the entire experiment is 45 minutes long, and there are many such replicates. So what you are seeing here is the following. On the top figure, you are seeing the polarization for group size 15. In the bottom two panels, you are seeing polarization for 30 and 60 fish individuals. Visually, you can already observe a whole bunch of patterns. The first one is that the group size N15 seems to be polarized in general more than these two, the higher group sizes. Secondly, there is a lot of stochasticity in the data. The time series is fluctuating fairly rapidly over time. The polarization is rapidly fluctuating over time. Okay, this is the experiment we have, right? But I mentioned to you one of the questions that people have been interested is, okay, we have all this collective motion, that's great, we can now quantify them, that's also fabulous, right? But ecologists and often want to understand what is it that each fish is doing, what is each bird doing. Can you tell me what are the individual level rules that produces these patterns? Okay, so how can we derive these rules from the noisy data that we have? Okay, so to do that, I have to explain some theory to sort of, we have this data, how do we analyze this data? For that, I have to now go back to some theory. So what's the theory? So I'm not going to go into a lot of mathematical details. I'm going to hide the mathematical details. Okay, so what I am going to show you is the following. Look at these two graphs, one on the left side, one on the right side. What are you seeing? You're seeing y-axis polarization. Okay, before I go, are there any questions at this stage? I can take any questions if something was not clear about the motivation or anything about what I've said so far. There is one question in the chat box. Oh no.
No, there is no question. It was me.
Okay. Okay, no problem. Yeah, before I go further, I can take any questions if there are any.
Yeah, if anyone wants to raise a hand, let me check.
No, so I can probably go forward.
Hi, sorry, I have a question.
Sure.
I apologize because I didn't follow last lesson. I couldn't be there. So maybe my question is redundant. By the way, like when you take in considerations like this kind of behavioral models, like of fish is moving, sorry for my words, do you take in consideration also the environment in this modeling, like let's say current, or your aim is the one to isolate only the motion of the group?
Yeah, that's a great question. So in this part of my talk, I'm going to assume that the environment plays no important role. So you're right that imagine if I was studying bacterial motion in their medium, the environment actually is really important. The viscosity of the fluid will be very important, right? Whereas when fish are moving in water, often it's not very important. The self propulsion is sufficiently strong, okay, that maybe the fluid dynamical effects can be ignored. Okay, so but I could be easily wrong because I'm not aware of there is only one recent study that looks at 2 or 3 fish and a fluid dynamical effect. I haven't read that very carefully, but I'm not aware of any study that has quantified this effect as of today. That's a great question.
Thank
As
you
of
very
now,
much.
I'm going to ignore that. Okay.
Thank
When you
you.
say environment, there could be other aspects of environment, right? Imagine
Of course.
there's a predator, right? That's also environment for the fish. It's an ecological context. That also plays a role on how they move. That is something, if I have time, I will cover in the second part of my talk.
Thank you very much. Cheers.
Okay. So, okay. Any other, if there are no other questions, I will move forward now. So, okay. So how do we analyze these data? I have computed this polarization as a function of time, right? How do I analyze these data now? Okay, that's the basic idea. So now I'm showing you 2 graphs here. What I have done here is I have plotted the polarization as a function of time. And as you will see, the polarization is varying between -1 to +1. Ignore the fact that there is a negative sign here. Trust me that that's not very important. But what you are seeing here is that these 2 graphs are pretty similar qualitatively. But but how did I get this data? I actually got this data from computer simulations of 2 very different types of models. Okay. But they look very similar, right? They are fluctuating between values of +1 to -1, right? And there is a lot of stochasticity. And if you plot the histogram or maybe the normalized density functions for the polarization, you find it's bimodal, meaning the value of m is either at +1 or -1. Okay, by the way, let me just tell you what is +1 and -1. What we were doing in the simulation was we were assuming that the world is a torus and animals can move only in one direction or the other direction. So if everybody moved in the direction, let's say counterclockwise, the map polarization will be +1. If everybody moved in the counterclockwise, the polarization would have been -1. Okay, so that's what I'm plotting here. Okay, so the histogram of these 2 look pretty similar. Of course, they're not identical. There are some differences, but the differences could be entirely quantitative. There's nothing fundamentally different between the 2, right? Okay, in fact, let me now tell you what was different between these 2. So the left column, I produced these values of polarization, assuming that each fish in the model interacts with only one other fish and copies the direction of motion. And then every fish keeps on doing this. That's how I produced this left column data. On the other hand, in the right column, I assume that each fish looks at 2 other fish and then it moves in the direction of the average of the other 2. Okay, approximately speaking. So as you're seeing in the left column, there is only a pairwise interaction. One fish looks at one other fish. There's a pairwise interaction. On the right column, the fish, there is a ternary and a triadic interactions. There are 3 fish and they all move in the direction which is the average, which is the average direction. They both produce very similar time series of polarization and they both produce similar PDFs. How do I know if I only had time series? How do I know where it came from, right? So to address this question, let me now describe the model in more detail and I'll tell you some results from the model. So what is this pairwise interaction model I just showed you? In this pairwise interaction, imagine there are a large number of fish, okay, and then you just pick 2 fish randomly. So you pick basically a pair, arbitrarily chosen pair, and then one of the individual will turn towards other at a rate c. Okay, so this is a probabilistic interaction happening at a rate c, and one of the fish will turn towards other at rate c. So as you can see, after this copying interaction, both the fish are moving in same direction. In this, there's an additional rule because we want to have some randomness in the system, which makes sense. A fish turns randomly by angle delta theta at a rate s. Okay, so there are basically a stochastic turning and a turning caused by copying a randomly chosen individual. This is what we call pairwise interaction model. Now I will discuss what is called the ternary interaction model, the right column of my simulation results. So in addition to pairwise and stochastic, we have those two. We continue to have them. We have now added a new rule called ternary or average interaction. So now what is this? How do we implement this? Look at this. There are three fish at the bottom here, right? On the left side of the bottom, where my mouse is moving. So this fish, which is turning away from the slide, is most disoriented in this triad. So what this guy will now do is turn towards the other two. So for example, the result of this ternary interaction is that this fish who was moving away from the slide is now moving towards the two other fish. So in some sense, this is causing an average effect. And that's why we also call it average interaction model. So unlike the pairwise interaction here, there are three fish involved in an interaction. And this happens at a rate h, which unfortunately I forgot to write. So the rate of interaction is h here. So I have described two models now. So what do we do with these two models? What we do is, we construct analytical model, and we try to look at that same quantity I showed you in the empirical world. In the empirical world, what did we do? I calculated this capital M, and I'm denoting here by small m for the models. The small m here is basically the summation of the velocity vectors of all the fish. Okay, so it's basically the average of the velocity vectors of all the fish. Okay, so we now want to understand what is the dynamic of this quantity M. Now, I'm not going to show you the mathematics. So it involves, as you can see, the rules for stochastic, right? And what we basically do is use ideas of master equation and something called system size expansion in the physics literature. And from that, what we do is we calculate what is a stochastic differential equation that describes this group polarization. Okay, so all the mathematical details are present in this citation. It's also available on the archive, so the physics archive. So you don't have to really buy this book. So what we find is the following: if the pairwise interaction model is correct, the dynamics of this M follows a stochastic differential equation given by this. So I know there are lots of terms here. Let's and then if the for the ternary interaction model, we find another equation which is again SDE, which is given by this. Now, what should you focus on? There are so many terms, right? Let's focus on the deterministic term. Okay, so even before that, let me point out that there is a stochastic term eta t here on the right hand side. Okay, on eta t is basically the Gaussian white noise. That's a stochastic term. And then we also have a deterministic term, which I have circled here. Now let me just try to see if okay. So we are focusing on this deterministic term. Clearly, you see that the deterministic term in the pairwise model and in the ternary interaction model are different. The pairwise interaction model has a simple linear term AM, right? Whereas the ternary interaction model, in addition to the AM term, also has a cubic term. Okay, and this H is the ternary interaction rate, which was not present in the pairwise interaction model. Therefore, basically, if we can compare the deterministic part, if you can somehow compute the deterministic part of your data, then you can distinguish whether your fish are following a pairwise interaction model or a ternary interaction model. On the other hand, you can also look at the stochastic term. Okay, the stochastic term again has a similar form. They are both quadratic in M. However, looking at the stochastic form here alone is insufficient because they are both quadratic. So one has to look at a combination of deterministic and stochastic term to distinguish these two models. Okay, so that brings me to the following question. We had, okay, before I go further, I want to say something. Okay, something about bistability and stochasticity. Okay, So you remember that in the previous talks on tipping points, I discussed this classic picture of a ball in a potential well. I think a lot of things moved. Oh, I don't know how it moved. Okay. Just give me 1 second, please. I'm trying to erase this first. Oh, something happened. I don't know what happened. Okay, I think it's fine now, but I was hoping I can write, but there is some problem. Okay, we remember we discussed this picture of ball rolling in a potential, right? If there are two wells in a potential, because of stochasticity, ball can move back and forth. And then if you now plot the time series, you find that system will go back and forth. Okay. On the other hand, imagine another system where you have a single well. The ball is typically, what do you expect if there is stochasticity? What do you expect? That it just fluctuates around this mean value, right? That's what you typically expect. However, whenever we say this, we often make an assumption that the stochasticity is constant for all values of the state variable. Let's go back to the previous slide. Look at this. There is a deterministic term, which is simply a exponential decay. There is a stochastic term, which is not a constant term, which actually has M dependence. So let me write down the equation again. So what we really have, so typically when we study many systems under a linear approximation, we do something like this. There is a exponential decay of the system from many perturbations. Therefore, m is equal to 0 is the fixed point. And then you have a stochasticity that makes a system go around the equilibrium value. What we have in the pairwise interaction model is something very interesting. What we have is this stochastic term is actually not a constant term. It actually has a m square dependence. What does that mean? What this means, when m is equal to 0, deterministic term is 0. However, the stochastic term is actually maximum, a plus 1 in this case, the way I have written it down. Imagine when m is now close to 1. If m is 0, right, the stochastic term is basically a plus 1, the way I have written. On the other hand, imagine m is equal to 1, which is the maximum order parameter, right? Maximum value of the order you can have. Then the stochastic term will be actually minimum. So meaning when the system is disordered, which is m is equal to 0, we have least amount of stochasticity. When the system is ordered, we actually have lowest amount of stochasticity. So this noise is playing a very interesting role here, because when the system is highly disordered, the noise is very high, and therefore what the noise then does is it actually kicks the system out of the zero equilibrium and produces a pattern like this. Okay, so the pairwise interaction model basically has a fairly counterintuitive effect of stochasticity. So it's also okay. So with that description, let me now come back to the fact that we have this two time series now. One of them came from pairwise copying, the other came from ternary interaction, and they both look similar. So if we can somehow construct a stochastic differential equation for these data sets, then I can distinguish where the data came from. So how do we do that? So there is indeed a method that actually can be used to construct where the data came from, and the basic idea is as follows. So the deterministic term basically is the average change in the value of the order parameter. On the other hand, the stochastic term is basically more like at the second moment of the change. Okay, so basically by computing the first moment of the change, it's called jump moments. First jump moment will give me the deterministic term of the SDE, and the second jump moment will give me the diffusion of the stochastic term in the stochastic differential equation. Okay, and in fact, if you apply this method, here is what you find. Here on the right hand side, now focus on the right hand side. If I use the data from the pairwise copying model, if I use the time series, and if I apply this formula, I do actually find the deterministic term is a linear term. On the other hand, if I apply the method to a ternary interaction model, the black solid line is the answer here, by the way. We find that, in fact, this term is actually cubic. And by comparing the fact that these two functional forms are so fundamentally different, we can infer that this data therefore came from ternary interaction, whereas this data came from the pairwise interaction. Okay, so that's the basic idea and theory. I will now stop for a few seconds and let you ask any questions, because some of these theoretical concepts may not have been well explained.
So if there is any question, there is no question in the chat, but
okay. There are no questions. Okay, fine. So I will go ahead and okay, there is one question.
There is a question from Tuan who is asking, it seems a kind of kinetic Ising model, right?
Which one of them is a kinetic Ising model? This is really closest to so I think the pairwise model is more like a water model. Okay, the ternary interaction model is more like a kinetic Ising model, because in Ising model you basically have the Jij sort of, you look at the not just one individual, one spin and turn, right? We actually often do a average of the neighborhood of the spins. So that's more like the ternary. That's my understanding. But in some limit, you're right. They're actually more like XY model, because also if you think of it in 2 dimensions. So basically, whatever I showed you here was all in 2 dimensions. All the vectors were in 2 dimensions. So all the analytical results are also from 2-dimensional. So all these all these M vectors are actually 2-dimensional vectors. So they are really more like XY, kinetic XY model with different interaction rules.
There is another question from Rajwal, who is asking, the criteria to distinguish between interaction rules come from top-down approach. How well does the criteria apply to bottom-up approach, where you start off with observational data and then try to plot the jump moments? Yeah,
that's a great question. And I think I have clearly done a very poor job of explaining this slide, which I have currently, I'm showing now. So the point of this slide is actually exactly that, which is, sorry, I think my slides went off. Okay, so that's precisely the point of this slide. I showed you, if I have a model, I showed you how to get the equations, right? What this slide is showing, and again, I forgot to cite the paper here. We have a paper early this year that shows this. What we have shown in this paper is the following. If you gave me only this time series data, which could be observational data or simulational data, here I am demonstrating for the simulational data. If you had given me these 2 time series without the labels of pairwise and copying, I would then apply this formula of capital Fm, and then I can construct the deterministic term for both of them. And then by comparing deterministic term, I can conclude that the pairwise mod, the left column came from pairwise because it's a linear term. The right column data came from a ternary interaction model because it's the data, because the function is cubic. Did I explain that point? So this is the bottom-up approach. So given data, how do you construct the jump moments? It's precisely this. And there are a lot of subtle aspects about this. What's the right time scale to choose? If you noticed all the equations here, the time scale over which we calculate jump moments, the first jump moment and the second jump moment are not same. How do you find all of this? This is in a paper which I hope I have put the citation somewhere in the end. Any other questions?
No, there are no other questions in the chat. Okay. Sorry. Yes.
One more. Sure.
There is from Bert, who is asking, is the system size expansion a mean field model with stochastic term?
Absolutely right. So the mean field model, mean field part, deterministic part is identical to mean field approximation. The stochastic part comes from the fact that the number of individuals in a fish school are finite. And that comes, system size expansion tells you how to construct the functional form for the stochastic term, also accounting for the fact that there are only finite number of individuals. So if n was infinitely large, that term becomes 0. So I'll just show that again. Oops. This one. So you can see that the stochastic part has 1 over root n term. So if n was infinitely large, stochasticity becomes 0.
Great. Okay. I think you can move on.
There's another question. I can take that up later again. I think it's related to the previous ones. But I think this is a general question. This is a great question by Prajwal again. Maybe at the end I can take this. Is that okay, Prajwal?
Yes, I think we can move on and answer.
Okay. Okay. Sorry, where was I? I was in this slide. So now I have just showed you the theory and in principle how a method can actually help us extract these jump moments from data. Okay. Now I had already shown you experiments, right? So I had already shown that we were able to compute these jump, order parameter polarization as a function of time. Okay. For different group sizes. Can you apply the method of jump moments and construct the stochastic differential equation for these data? That's precisely what we did. And so this is the, okay, I have already shown you the result now. What we have done, we did exactly that. We did this for all the 3 group sizes simultaneously, and then we chose the best fit model. And this is the equation we found. And as you can see, the deterministic equation we found was actually linear. The stochastic term we found has, again, this mod m square behavior, which was exactly same as that of the pairwise stochastic copying model. Okay, it almost seems magical. It's actually true. So what we therefore infer from our analysis is that because the deterministic term does not have cubic terms, and the stochastic term also matches with the qualitative features of the pairwise model. So our fish, which we analyzed, must be interacting with one another using the stochastic pairwise copying interaction rules. Okay, and moreover, not only that, we also can rule out the fact that ternary interactions are dominant mode of interactions. So, I mean, of course, I think I should have been careful in writing this second bullet point. We can never really rule out what fish are doing. What we can say is whether they are dominant or not. So clearly it appears that the ternary interactions are not dominant. If they were dominant, we would have found traces of cubic terms in the deterministic term. So for those who might be interested, I can, the sort of, if you look at the goodness of fit kind of measures, one of the measure is called R square, and the R square value for the this model is close to 0.9, which gives us a very strong sort of, reason to believe that this is a statistically good fit. Okay, so again, just to remind, pairwise interaction model had these 2 very simple rules. There was a stochastic turning rate, right? And then there was this pairwise copying rate, where one fish just looks at one other fish and turns towards it at a rate c. and this rate C and S become part of the stochastic differential equation in this very simple way. Okay, so to summarize this part of my talk, so what I have shown you is that fish that we studied actually show simple pairwise interaction, pairwise stochastic interactions. This is quite contrast to classic models of local averaging. For example, the Vichek model. For example, the Vichek model that I mentioned, that many of you might be familiar with, assumes that each self-propelled particles looks at certain unit radius and looks at the direction of every individual in that radius and finds an average and moves in an average with a small error, right? That's the Vichek interaction rule. Likewise, the cousin model also does something similar. It also has rules for attraction and repulsion. Okay, now in our analysis, we were focused entirely on alignment only. So I can't talk about what's happening in the domain of attraction and repulsion. But if you focus entirely on the effective alignment rules, what we find is that it's contrast to the classical models of local averaging. The fish seem to be doing actually even simpler than what the Vichek model actually says. And we also, provided a method to construct models from the data. If you have a high resolution time series data, you can actually construct a stochastic differential equation model from those data, time series data. And these are the 3 references for those who are interested. They're also all easily available from my website, the PDFs or the archive links. So for somebody who's interested in understanding how do you go from these simple interaction rules to stochastic differential equation models, this chapter provides the review of the theory and some methods. It's more of a pedagogical article as a part of book chapter. And then this paper, the second paper, tells you how do you go from this bottom-up question. If you had observational data, how do you go towards the theoretical? How do you construct an SDE? And the third paper actually applies both of them, and to the specific fish schools we studied. They're called Etroplus or Karimine locally. Okay, so that's the summary of the first part of my talk. Do I have more time to cover one other ongoing work on collective behavior?
Sure, absolutely. I mean, we have time. You have still 10 minutes, but we can go a little bit beyond.
Okay, now let me present another work where we look at how collective motion happens and sort of responds under the context of predation. You may remember the video I showed in the beginning where there were really fascinating escape patterns of fish schools, starling flocks, and I think krills responding to predation. Okay, so if you think of understanding collective motion in nature, they're not clearly in a fish tank of the type I showed, right? Secluded, free from any sort of threats and so on. So there are while many of the social interactions do shape collective movement in the nature, there are habitat features, environmental features, and importantly predation that shapes many of the interactions between animals that in turn causes collective movement, right? So much of the work in the field has sort of focused from the middle box to the right side arrow, and the focus on the left side hasn't been as much. And to understand this, one of my students is looking at this beautiful charismatic species called blackbuck. These blackbuck live in grasslands, and they are native to Indian subcontinent. The males are the ones with dark coat and these beautiful horns, and females are the orange bodied and typically slightly smaller in size. And many of the herds in this species are female groups with a few males here and there. Okay, and they move from place to place. Many groups merge together. So there is no single group that maintains identity as a function of time. So they keep merging, splitting, and moving around in the grasslands. Okay, so what we did was, of course, to observe a natural predation event, and then being able to record high resolution data is very, very difficult. So what we did was the following. We flew drones at a height of 30 to 60 meters over the herds, and we record them for about 15 minutes. Midway through the recording, one of the students or the experimentalist who is doing experiment will approach this herd. And these are really shy animals. And what these animals then do is observe this human coming towards them, and they escape or move away from this approaching human. And this approaching human is moving very slowly, cautiously, to not to make sure that it's as less invasive as possible. So this is a sort of a perturbation to the natural state of the groups of black buck, but it also acts as a mild threat. So this could be, we thought we could use this to understand how animals respond to perturbations and threats. So here is an example of a video. I hope it's playing. I hope the resolution is okay for you also. So this is the drone footage. As you saw a few seconds ago, somebody from the bottom part of the video moved towards the center, and there is now another part of the herd from the top right where they're all moving towards the rest of the herd, and now they have all picked up a bit of a speed and they're all moving in the same direction. Okay, all of this happened because there was somebody who one of the experimentalists moved towards this herd from the right side. So 30 seconds to 1 minute of this clip, although the entire video is, we have recorded about 7-8 minutes before this event happened, and also how does this event really settle down? How do herds settle down? Okay, so what we basically do is, with this data, we again use various image processing techniques, and we quantify the group dynamics, group cohesion, group polarization, group speed, and how do these groups respond to this simulated perturbation, simulated threat. For example, who initiated the response? Are there leaders and followers in this response? So what we basically do is we plot the two quantities here. I'm showing you two quantities. So in the herd, we first plot a mean near neighbor distance. If this mean near neighbor distance is small, that means the group is very cohesive. If it's large, that means the group is now sort of spread out. So we plot time series of this, and then we also plot the group polarization, how aligned they are, with one another. What we do based on these time series is we can clearly identify that there are 4 events in this time series. One is the pre-perturbation event. There is a initiation of escape, and then that in turn causes coordinated movement, and then there is a settling of the behavior. So those are all drawn using this red bars here. You need to use combination of near neighbor distance and polarization. You can see this two very clear bouts of coordinated motion. You can also see a bout of increased distance between each other when they were responding. Okay, so we have done this for about 60 to 75 videos, and the results I'm going to show today is only for a smaller number of them. So what we want to understand from this is how did the information about the perturbation or the threat, how did that spread across the entire group? And that we try to do by looking at how does the change in the speed of individuals happen from one individual to the other individual. And what we do to quantify this, what we do is we first identify who are the initiators, who are the first to respond, and we identify the initiators by basically looking at the speed of among all the individuals whose speed suddenly changed, and that individual is the initiator. And then the laggers, who are the last to respond, and from the initiator to laggers we sort of construct a network of how the influence is propagating in the entire network of these individuals. Okay, and secondly, we also look at, are there some individuals who are more influential than the other individuals as individuals are moving in the entire group. And finally, there are also individuals who perform movement sometimes irrespective of what everybody else is doing. They are called isolated individuals. So here is an example of analysis and the results. So the approach was from the from the towards north, and this was the group. The numbers here show the order in which they responded. So as you can see, there's a lot of counterintuitive facts here. One is that who is the first individual to respond, second, third, fourth, they're not necessarily very obviously determined by the spatial locations of animals and where the threat is coming from. And then what we basically therefore do, this is somewhat systematically, we look at the pairwise cross correlation between individual velocities for all pairs in the entire group. And then for each pair, we identify who was ahead of the other in the velocity changes. So the basic idea is that, if you look at pairwise correlation in the directions, if one individual is always turning before the other individual turns, you can assume that the first individual is then the leader of that pair. So we do this for each pair in the entire group, and we sort of rank the strength of this relationship by the strength of correlation. So I'm going to show you two examples here. For example, if there are three individuals, A might be influencing C, C might be influencing B. In which case you say A is the leader, C is the second, next follower, followed by B, right? On the other hand, it could be more complex. A might be influencing C and B. C is influencing only B, in which case A influences C, influences B, but A also influences B, right? So you can now construct a network and the strength and a network where the nodes are individuals and the arrows and the strength of arrow represents how strongly they are influencing each other. Okay, so here are the four networks that we find corresponding to four different stages of the experiment. This is the pre-perturbation. As you can see, the network really has no structure. Everybody is sort of independent. There are no clusters in the structure. And then there is an escape bout, right? And in escape bout, there are some that are initiating the movement and then they become correlated with the others. And those are all here in this greenish, this blob here. And then finally, when they become coordinated motion, you can see clusters. There are sort of small clusters that are moving in a coordinated ways. And then when there's when they all sort of come to some kind of a post-perturbation equilibrium, it again looks more like the pre-perturbation event. So we can actually construct these networks of how the information is flowing. And I'm going to just summarize what are all the results we have found, very preliminary results. These might change when we analyze more rigorously. One is that we basically find that the group as a whole speeds up first. It's so imagine you are an animal in the group. Should you first orient with your neighbor or should you first run? This might be one simple question you can ask. What we find from this analysis is that the groups usually first speed up. As a consequence, they seem to orient with one another. Okay, so and the third effect usually is that of being more cohesive. Because they're speeding up and orienting, then you find that the group is now more cohesive. Okay, and then secondly, we find that there are these leader-follower relationships that happens only in this escape event and not in the other three events. And finally, there is a lot of variation, between individual responses. For example, the ones who initiate the response are not necessarily same as the influential individuals. The one who has most influence on the movement of others need not be the ones first to respond. So that's another interesting results. So these are all some preliminary results of how we can understand collective motion and pattern formation in the natural conditions. So that's sort of the end of my talk. So the work I presented today, the first work, was done by PhD student Jitesh Jawar, who is now a postdoc at Max Planck Institute in Constance. And the second part of the work on black buck is done by Akanksha Rathore. And of course, there are many collaborators involved, like in any other project. So that's the sort of end of my talk today. Thank you, everyone. And I can now take more questions.
Great. Thanks a lot, Vish, for the very nice talks. So there was the question by Prajwal
Yeah.
that perhaps we can address now. I'll read it so people in YouTube can know it. So Prajwal is asking, what about cases where you don't know what the average rules are? So the previous question was, sorry, about distinguishing between interaction rules coming from top-down approach and sort of the connection between theory and data. And this question is, but what about cases where you don't know what the average rules are? I guess I'm thinking more about the Vishwesha model and whether the jump moment still remains cubic or changes to something else. When looking at the data, we wouldn't know if the first jump moment will be cubic or linear or something else.
Okay, so so Vishwesha model also has a cubic term. So if you look at the mean field Vishwesha model, it's exactly same as a ternary interaction model, which is why I was interchangeably using ternary and Vishwesha, ternary and average. Ternary also has an average interaction model. So, but you're right that, okay, more generally, if there were some complex interaction rules for which you cannot analytically determine such a closed form solution for the jump moments, what do? I don't know. But that problem is a very universal problem, right? So see, there is no way we will ever observe what any individual or cells or animals are doing, right? We're only going to look at some coarse grain property, and we measure that, and then measure something about the coarse grain property, right? And then we see which of the models best explains this coarse grain property. And in this case, it happens to be the stochastic differential equation itself. So yeah, so I hope I don't know if my answer is very clear either. You're right that when we're looking at data, we wouldn't know, but we can construct from the data, as I mentioned, right? I hope that answer was clear. Now, how do you construct equation from data? Was that clear, by the way? Because that's very central to what I said. Because if you cannot do that, there is no way you will be able to distinguish the models.
Yes, there is no sign in the chat from Prajwal, but I guess it's a sign that it's clear. So we have time for another question. If anyone, okay. Okay, there is another point made by Prajwal. I think you can answer these and then we can move forward. So the previous answer is clear. Thanks. I have a set of questions. Smiling face. One, in the models, you show that most other models, the speed is considered constant. But in the black buck case, that's not true. How will this, changing this hypothesis change the model and the dynamics? So then there is another question. In the same group, if you repeat the perturbation multiple times, do the leader individual and the hierarchy stay the same? And in line with the previous question, if repeating multiple times, how would one control for all sorts of different effects like memory or learning? So I guess the question can be summarized in whether it's true that the speeding constant and the hierarchy is constant.
Yeah. So you're right that in the simple models, the speed is constant. So there are some groups which have developed variable speed models. We also have developed one that is sort of driven by the data we have. And it does show some surprising results. So it depends on the question you are trying to address, whether the variable speed is important. For example, even the fish have variable speed. Right? It's not that they are always moving at constant speed, right? But if the question is the polarization dynamics, maybe it's not very important. Okay. But if there's some other question, in the context of prediction, speed does become important. So therefore, in such cases, one has to certainly change the model. I fully agree with you. I mean, that's also true for black buck in the sense that the very thing we are measuring really is changes in the speed. So coming to second and third questions, can you apply this perturbation multiple times to the same group? You're absolutely right that the learning will play a huge role when you do this. So therefore, we don't apply perturbation to the same group over time. So we always try to ensure that we have found a different group and we don't disturb the same group repeatedly. So memory, a lot of other factors play a role if you repeat the, the same experiment or the same group.
Great. So I think we can stop here. I don't see any other question. So I'd like to thank you, Vishu, again for the 3 great lectures. And thanks all the participants for their participation. So the next session is going to start at 2:30 Italian time. So we have more than 1 hour to take a break from the screen. And then we'll be back with 2 panels and the last 2 lectures by Justin Nickel and Alvaro Sanchez. So thanks everyone again. Thank you, Vishu. See you in 80 minutes.
Thank you, everyone, and thanks for the invitation, Jacob.
Thank you.
Bye. Bye.
Just a technical information, the Zoom meeting will stay open, I guess. So if you want, you can stay connected. But you can also disconnect and reconnect.